So we'll begin by discussing about the many types of cells which constitute the central and peripheral nervous systems in our body. So these are the neurons and the glial cells. So these are basically scaffolding or supportive cells which mainly aid in synaptic interactions. And these can be divided into astrocytes, oligodendrocytes, and microglia. So now we'll see what a singular neuron actually looks like. Structurally, we can divide the neuron into three cellular compartments. So first of all, we have the cyton or the cell body. The cell body extends into further projections, and these projections are called the dendrons or dendrites. Okay, and the singular long projection in the opposite direction as that of the dendrons is called the axon. The dendrons or dendrites are basically tapering extensions of the stroma itself. So these have basically specific ligand-gated sodium channels or various types of ion channels which help the neuron to respond to other stimuli or other signals by other nerve fibers. Also, these may have secondary or tertiary extensions, as in secondary or tertiary dendrites. And the more the amount of branching of these neurons, more the amount of branching of these dendrons, more is the complexity or the severity of the information that is being transmitted along a nerve fiber. Then we come to the second compartment, which is the cyton or cell wall. So here we have the nucleus, which is the genetic center as usual. We also have the mitochondria, which is the metabolic center, and it also has some very specific functions in a neuron. For example, it provides the energy for synaptic transmission, and also it prepares the substrates for various synthesis of metabolites, neurotransmitters, etc. The mitochondria also helps in secretion of certain enzymes which help in degrading or diminishing the quantity of certain neurotransmitters according to the need of the body. For example, what monoamine oxygenase B does for 5-HTO serotonin and catecholamines, and what GABA transamine does for GABA. So as per the physiological needs of the body, these neurotransmitters are diminished well in time by the release of such enzymes in the mitochondria. So what else is there in the cyton? We have stuff like Michel's granules, rough endoplasmic reticuli, and the ribosomes, which all aid in protein synthesis. And then we have the axon. The axon terminates via specific structures called the synaptic knobs or blobs on either a neuron or a muscle fiber. Then the part of the cell body from where the axon extends or originates is called the axon hillock. So the outermost cytoplasmic layer of the nerve fiber is called the neuron. So what does it do? It basically helps in regeneration and quick regeneration of an already injured nerve fiber. Now, as this is a myelinated nerve, the mass needs to spread the Schwann cells which help the production of the myelin sheath. So in the peripheral nerve system, the Schwann cells help in secreting the myelin sheath, which acts as the insulation and also has various other functions, but it basically helps in insulation or prevention of loss of charges or energy of the information that is being transmitted along the nerve fiber. The spaces along the nerve fiber where the axon is devoid of the myelin sheath, these are called the nodes of Ranvier. And these are about 1 micrometer in length. The axon also has a few important specific features exclusive to it. These include absence of endoplasmic reticulum, absence of Golgi apparatus, and absence of free ribosomes as well. What may be present is smooth ER, which help in storage of calcium, intracellular storage of calcium. And by this it helps in maintaining intracellular calcium levels. If we have to broadly describe the functions of these cells as in being a part of the nervous system, we can include the obvious motor functions, the sensory functions, then processing of the information, and finally the storage of the information.